Okay, so welcome to this next video in which we're discussing the aldose monosaccharides. Okay, right, so we have now discussed the cyclization of D-glucose, okay, to form D-glucopyranose. Now I want to also show you the structure of manopyranose. So basically when you react the penultimate carbon's alcohol group with the aldehyde group, you get a six-membered ring like this, and this six-membered ring is known as a pyranose. Okay, so you can then also do this for mannose. Okay, so remember the only difference between mannose and glucose was that this alcohol group of the first carbon went in different directions, basically. So in glucose it came out of the page towards us, in mannose it goes into the page away from us. Okay, right, so let's now draw the structure of D-manopyranose. Okay, so everything is the same pretty much apart from that one change that we made where this alcohol group now goes into the page away from us. So let's start by drawing the ring, which is going to be within the plane. Okay, so here is the ring structure, the pyranose ring. Okay, and then basically, because we are talking about D-mannose, and this alcohol group here therefore comes out of the page, when you react this with the aldehyde group, basically this methylene group with the alcohol group coming off, this sixth carbon ends up coming out of the page towards us. Okay, so because we're talking about D-manopyranose, this sixth carbon comes out of the page towards us, like so. And then of course it will still have its alcohol group coming off. So we're still drawing skeletal structures, so we don't show hydrogens coming off there. So if we're talking about L-manopyranose, that sixth carbon will be going into the page rather than out of the page. Now, off this fourth carbon down here then, so which carbon was the equivalent of the fourth carbon? Well, it's this carbon here. Now remember, the only difference between glucose and mannose is that this alcohol group was different. It was going into the page away from us. So now this should be exactly the same as in glucose. Now it's coming out of the page towards us, but remember when we cyclize, everything turns it backwards. So this now goes into the page away from us. Okay, then the carbon off this, the alcohol group off this third carbon over here is then going to come out of the page towards us because originally it went into the page away from us. Okay, so it's coming out of the page towards us, and then now off this second carbon the alcohol group is going into the page away from us in D-mannose, whereas it was coming out of the page towards us in D-glucose. Okay, so when we cyclize, that means it's going to come out of the page towards us because everything is turned backwards when we cyclize. Okay, and then the alcohol group that you now have off this anomeric carbon, okay, which is the first carbon, that, remember, now has an alcohol group off, which is the transformed carbonyl group, and this can go either into the page away from us or come out of the page towards us. So I'll draw this squiggly line which denotes it can either go into the page or out of the page. Now if it goes into the page, that will be called the alpha anomer. Okay, so we'll call that alpha D-manopyranose. Okay, and if it came out of the page towards us, that would be called the beta anomer, and that would be called beta D-manopyranose. Okay, so that would be beta D-manopyranose. Okay, so both of these are optical isomers, which of course are epimers because they differ by the optical isomerism of a single alcohol group. Both of these are expressed within cells, so alpha D-manopyranose and beta D-manopyranose. The L isomers are not expressed, so the L isomers where this carbon goes into the page away from us, they're not expressed basically. Okay, and I shouldn't really say expressed. Expressed implies that they're a protein generally. Present would be a better word. Okay, so let's now turn our attention to the final important one, which is galactose. Okay, so we'll start by drawing the structure of D-galactose, and then we'll draw it cyclized. Okay, so here's a new piece of paper. So the structure of D-galactose then. Okay, so the alcohol group in the first position came out of the page towards us. then we had 2 going into the page away from us and 1 coming out of the page towards us. So let's draw this here. So here is the carbonyl group here. Here's the first carbon, the second carbon, the third carbon, whoops, the fourth carbon, the fifth carbon, the sixth carbon, and then the alcohol group coming off the sixth carbon over here. Right, then off the first carbon, the second carbon, I think of it as the first carbon, but obviously it's the second carbon, the alcohol group is coming out of the page towards us, so let's draw that here. So here, the alcohol group comes out of the page towards us. Then in the second the third carbon this is, the alcohol group goes into the page away from us. Off the fourth carbon, it goes into the page away from us. And then off this fifth carbon over here, it comes out of the page towards us. Okay, so this is the structure of the molecule Dgalactose. Okay, so now again we can cyclize this to make D-galactopyranose, and again there's 2 anomers of D-galactopyranose, the alpha and the beta anomer. So let's show this. So the process is pretty much identical. All that's going to differ between the different pyranoses is the position of the alcohol groups of these 3 here, because that's determined by the name basically. The position of the alcohol group, the position of the groups of this carbon and this carbon are determined by the alpha, beta, and the DL isomer naming system. Okay, right. So here is the ring. Now, of course, because we're talking always about the D isomer, this 6th carbon comes out of the page towards us, and it will have its alcohol group here. Okay, now the alcohol group of the 4th carbon here, that's the 4th carbon here. So that was going into the page away from us. Now remember, everything's turned backwards. So this now comes out of the page towards us. So in galactopyranose, this is going to come out of the page towards us. So we're now talking about D-galactopyranose. Okay, right. And I haven't specified yet whether we're talking about the alpha or the beta anomer, so obviously I'll put a squiggly line there to denote that I'm not specifying. Okay, so here is the alcohol group coming out of the page on this 4th carbon. Now the 3rd carbon, it's going into the page on the linear structure, so it will be coming out of the page on this pyranose structure here. Okay, so here it is coming out of the page. This 2nd carbon down here, it's coming out of the page on the linear structure, so it should be going into the page on this pyranose structure. And then off the anomeric carbon, you then have the squiggly line, because we don't know whether it's going into the page or out of the page, because I only said we're drawing the structure of D-galactopyranose. I didn't say specifically alpha D-galactopyranose or beta D-galactopyranose. So if you were asked to draw the structure of D-galactopyranose, this is what you would draw. Okay, right, now let's turn our attention to ribose. Okay, so this is different, because remember, ribose, D-ribose, was a aldopentose rather than an aldohexose. However, the reaction is pretty much the same, so let's just remind ourselves of the structure of D-ribose. So remember, it was this 5-carbon structure where all of these middle alcohol groups came out of the page towards us. So let's put that here. So, D-ribose. Okay, so the structure of D-ribose in the plane went like this. Here is the carbonyl group. Okay, that's the 1st carbon. 2nd carbon, 3rd carbon, 4th carbon, 5th carbon, alcohol group off the 5th carbon. Okay, now the alcohol groups of the 2nd, 3rd and 4th carbons are all coming out of the page towards us. So this is the linear structure. This is what, strictly speaking, is meant by D-ribose. Okay, right. Now again, what you can do is cyclize D-ribose. Okay, so you can react the alcohol group on the penultimate carbon, okay, so over here, with this aldehyde group over here. And what you're going to produce is something known as D-ribofuranose. Okay, so you're going to produce a 5-membered ring this time, rather than a 6-membered ring. Okay, again, it's a hemiacetal. Okay, and again, you need to specify whether it's the D or L isomer, which will differ, of course, because they're built from the D or L isomer of ribose. Okay, right. So, what's going to happen? Well, basically, you're going to end up with a structure that's a 5-membered ring, and you've got an you've got an oxygen there, okay, and these 4 carbons, and then coming off this 4th carbon of this ring here, you then have the 5th carbon, okay, which is this carbon over here, okay, and it's got an alcohol group off it. So, this whole structure here, this is this, okay, and you have reacted this alcohol group with the carbonyl group here, okay, and the oxygen that is in this ring is the oxygen from this alcohol group. The oxygen from the carbonyl ring has now formed an alcohol group that will come off this carbon here, and again this is referred to as the anomeric carbon. So the carbon of the aldehyde group is referred to as the anomeric carbon. So this is the carbon of the aldehyde group, so this is the anomeric carbon. Okay, and again there are 2 anomers. Either the alcohol group that you form from this carbonyl oxygen can go into the page away from us, or it can come out of the page towards us. So what if we aren't specifying whether we're talking about the alpha isomer or the beta isomer, we'd put this squiggly line here, and we haven't specified. We've just said D-ribofuranose, okay? So if it goes into the page away from us, that would be the alpha isomer, so that would be alpha D-ribofuranose. And if it came out of the page towards us, that would be the beta isomer, that would be beta D-ribofuranose. But we've just said D-ribofuranose, so we would put this squiggly line here. Now, what about these final 2 carbons, the 2nd and 3rd carbon, these 2 carbons here, that weren't involved in the formation of the hemiacetal? Well, remember, everything is turned backwards, so these 2 alcohol groups, initially they came out of the page towards us, but things have been turned on their heads. So now both of these alcohol groups go into the page away from us. Okay, so that is the structure of D-ribofuranose. So these 5-membered rings are known as furanose, basically. So, basically, in order to specify the exact molecule you're talking about, you would firstly have to state whether you're talking about the alpha or the beta anomer, okay? Then you would have to state D or L to specify whether this 5th carbon comes out or into the page, okay? And then the ribo here takes account of these 2, okay? And then the furanose tells you that it's a ring. Right, so that's the structure of D-ribofuranose. And of course, if you haven't specified whether it's the alpha or the beta anomer, then we can't know, so we put this squiggly line here. Okay, so that concludes our discussion of the aldoses. I just want to emphasize that you can cyclize all the other aldohexoses and also all the other aldopentoses to create pyranoses and furanoses. Okay, and these would have a different name, obviously, so you'd have a different prefix to the furanose or to the pyranose, and you change these 2 carbons alcohol group optical isomerisms in the case of the aldopentoses, and these 3 alcohol groups optical isomerisms in the case of the aldohexoses. Okay, right, so that now concludes our discussion of the aldoses.